Angiosperms are defined by their ability to produce flowers and bear seeds that are enclosed within a fruit. This internal protection is one of the primary characteristics that distinguishes them from other plant groups. To trace the development of these seeds, we have to look at the anatomy of the reproductive engine, the flower. In this cross-section, we isolate the two reproductive components. The male stamen consists of the filament and anther, while the female pistil contains the ovary. For reproduction, pollen travels from the anther to the stigma, growing a tube down the style to reach the ovules. By utilizing the flower's anatomy, angiosperms create a controlled environment where the ovules are never exposed to the outside elements during fertilization. Zooming microscopically into the ovary reveals the embryo sac, which develops inside the ovule. This schematic reveals the specific cellular configuration of the embryo sac. It consists of exactly 7 cells, but it contains 8 nuclei. At the bottom, we see a 3-celled egg apparatus, 1 central egg cell and 2 flanking synergids. At the opposite end, there are 3 antipodal cells. The final cell is the large central cell. It holds the remaining 2 polar nuclei, which eventually fuse to produce a diploid secondary nucleus. This 7-celled, 8-nucleate arrangement is the standard internal structure found in angiosperms before fertilization begins. Reaching the embryo sac, the pollen tube releases 2 separate male gametes into the internal chamber. The first fusion event is syngamy. 1 male gamete fuses with the egg cell to form a diploid zygote. Simultaneously, the second gamete fuses with 2 polar nuclei, creating a triploid primary endosperm nucleus, or PEN. Take a moment to recall, what do the zygote and the PEN eventually develop into? The zygote develops into the embryo, while the pen develops into the endosperm, which provides the essential nourishment the embryo needs to grow. Because this double fertilization ensures that the nutrient-rich endosperm only develops after the egg is successfully fertilized, the plant avoids allocating resources to ovules that do not contain an embryo. After fertilization, angiosperms are classified into 2 groups based on the number of embryonic leaves, or cotyledons, found in their seeds. Monocots have 1 cotyledon and a fibrous root system. Dicots have 2 cotyledons and typically develop a main taproot that grows deep into the soil. You can also identify them by their leaves and flowers. Monocots show parallel venation and trimerous flowers, while dicots show reticulate venation and flowers in multiples of 4 or 5. Internally, the vascular bundles in a monocot stem are scattered, whereas in a dicot stem they are arranged in an organized ring. These 5 morphological traits allow for the systematic classification of any flowering plant. On an exam, you may be asked to explain double fertilization and identify why it is unique to this group of plants. A complete answer must define syngamy and triple fusion, then explain that the simultaneity of these 2 fusions is the specific event that defines double fertilization. This map consolidates the data, showing the ploidy levels for each fertilization product alongside the morphological traits used to distinguish monocots from dicots. Connecting these microscopic reproductive mechanics with macroscopic traits provides a full structural and functional overview of the angiosperm life cycle.